That's why I went off with some colleagues, some oncologists and drug developers to found Syros, this young pharmaceutical company that we founded about 18 months ago. But I think there's much more of that. I think there are things we can disrupt that are quite common across a particular cancer type, no matter how complex. And I think there may be things we can disrupt that are critical for multiple types of aggressive cancers. And I think David's RAF protein is one of many examples of that.
I don't have too much more to add, but clearly the pendulum has swung, right? So from everything is the same to everything is different. And the truth is there are probably dozens of vulnerabilities now that one could go after. Not the millions of cancers out there are not all different in terms of what they care about, but they're quite different. It's simply not as extreme as the two ends that you put there, which is a good answer for us, right? Because if they were all very, very different, then we get back to this issue of how are we ever going to fund develop inhibitors to these things and targets, because as was been alluded to, it's an incredibly expensive endeavor to even develop one drug. So I think to some extent the answer is good news, that there is. When we first did those screens, we were actually really afraid that we might not find anything in common, that they were all unique. But there are. They're not enormous. It's probably between 2 and 10 things that you can pick for each type of cancer.
Well, just a quick question about commonalities in cancer. I would like to hear your opinion on what is called shortening of the telomeres. Telomere shortening that's been, reported in pretty much every cancer. What's the relevance?
Well, actually, as you age, your cells seem to have shorter and shorter telomeres. Telomeres are the ends of chromosomes. All our chromosomes are linear and have ends, and there are these repeated sequences on each that help maintain the integrity of the ends. And as your cells age, the telomeres get a little bit shorter. Cancer cells, because they're going to rapidly divide, need to fix the problem of shortening telomeres, and so they end up generally activating ways of making longer telomeres.
Yeah, in fact, people have argued that the enzyme that makes telomeres, called telomerase, which incidentally Bob Weinberg also discovered, would be an interesting cancer target. The problem is it actually takes quite a bit of long time once you inhibit that enzyme to get the telomeres to shorten back down to where there would be a problem. You'd also probably wipe out your gametes and your stem cells, so it's probably not going to give you a selective inhibition.
Are you able to speculate on the impact that this approach would have on two of the major issues that concern people in the cancer community, which is time to bring a drug to the market and pricing? Can you say anything about how you think this approach could impact those two major issues?
What was the second issue?
Pricing.
Pricing. Oh, I'm an expert on pricing. Well, let me address the first just by telling you, a personal story, and that is about 3 years ago I got involved with a medicinal chemist at Harvard named Nathaniel Gray, and he was developing a small molecule chemical probe, not yet a drug, against a cyclin-dependent kinase called CDK7, and we worked with 5 different Dana-Farber cutting-edge cancer physician scientists and found that this small chemical probe had a profound effect on the tumor cells that they were most interested in. And so what we did was, being excited about that, we formed a company called Syros. In 18 months, it raised $85 million. we hope to be in phase 1 clinical trials in 6 months. That's very fast. It's remarkably fast to go from a chemical probe to a drug, which is what Syros has done, to a phase 1 clinical trial. Pricing really is, way out of my expertise. But, I think if it takes less time to develop something, you no longer have this argument that it cost you $1 billion to get to that molecule. So presumably, I don't know that pharmaceutical companies will price according to this logic, but if it's true that it only takes 3 years to go from concept to a drug molecule in phase 1, that should reduce the cost of developing these types of compounds. Now, that drug has not proven either safe or efficacious in humans, so you asked me to speculate, and that's my speculation.
Okay, let's thank Rick and David and Hazel again for their presentations this morning. Okay. And I'd like to, again, express my thanks to all of you for your support of what we're about. You give generously, you make introductions on our behalf, and you spread the word about our research. And so in parting, and you're very welcome to stay for conversation afterwards, I say we're not really parting. We leave with an invitation to join us on the journey ahead. So thank you all very much for coming this morning.